So, welcome back. Last session, last full session. We are determined to start now sharp. And I have the pleasure to introduce the session for you. It's a session devoted to plant clinical trials. And we are going to see an entire range of approaches, which in my book is great. That ranges from advanced therapies, gene therapy, to over ASOs, to small molecules. And antibodies will be covered as well. So I am excited that we can discuss together these upcoming research opportunities. And without further ado, I have the pleasure to introduce the first speaker from AskBio, Nathalie. And she is going to discuss with us restoring brain cholesterol metabolism. And she's using gene therapy for it.
Hello, hello. Thank you very much for the introduction and for giving me the opportunity to present our clinical protocol and on gene therapy addressing the brain cholesterol metabolism to treat Huntington disease. So just to introduce you to AskBio, it's a gene therapy company based on AAV delivery. We address rare and complex disease. We have 2 clinical trials, as you see, ongoing, and we are preparing this Huntington disease clinical trial addressing the cholesterol metabolism. So the dysregulation of brain cholesterol metabolism has been well characterized in human patients, but also in different models of Huntington disease. And as you see here on this slide, it's the dysregulation of this cholesterol metabolism is really directly, strongly implicated in the different hallmarks of the disease that finally lead to neuronal death. Brain cholesterol is extremely important. It's an important compound of our brain for 2 reasons. One is it's a major component in the membranes, and increasing cholesterol content in membranes makes the membrane sticky, decrease the membrane fluidity, and has major consequences on synaptic activity, plasticity, and ultimately nerve cell survival. But also neuronal cholesterol has a very important effect on cell metabolism, and particularly on the production of ATP by the mitochondria. And you know that ATP is extremely important for neuronal function. Neurons are extremely demanding cells in terms of energy. So at the end, this leads to stress accumulation and neuronal death. So what is very specific, all our cells produce cholesterol, of course, but what is very specific for the brain is that it is not permeable to cholesterol. So cholesterol has to be synthesized in situ in the brain, and when it is in excess, to maintain the balance, it has to be transformed to be excreted out of the brain in a compound that can cross the blood-brain barrier. This compound is a 24-hydroxycholesterol produced by an enzyme called 24-hydroxylase, called CYP46A1. This enzyme CYP46A1 is extremely important, not only because it's doing this and allows to excrete excess cholesterol out of the brain, but also because it activates the whole pathway. And as you see here, this pathway produces a number of compounds that play a critical role in neuronal functions, particularly vesicular transport, endocytosis, autophagy, and I will come back a lot on autophagy later, and synaptic transmission. So really this enzyme is key, it's neuron specific, and it plays a global role on brain metabolism. The 24-hydroxycholesterol product from this enzyme is decreased in the blood from Huntington patients. This has been published in several papers. We have, with Alexandra Dur at Pitié-Salpêtrière, also demonstrated in a cohort of early patients from the French Paddington cohort that indeed 24-hydroxycholesterol is decreased in the blood of these patients, cholesterol, total cholesterol being normal, and that this decrease is correlated to the UHDRS score among patients with early HD. So definitively, this 24-hydroxycholesterol is a biomarker of Huntington disease and can be used as a marker of target engagement in any therapeutic approach. The enzyme CYP46A1 is decreased in patients' brain and in different mouse model brain. This has been well described in the work we have done with Sandrine Bétuin, and she's here in the room today, demonstrating that in patients' brain, but also in the R62 mouse model where you see that the protein is decreased. So when the protein CYP46A1 is decreased, not only this has two consequences. Cholesterol accumulates, and particularly in membranes, as I said, so leading to sticky membranes and to finally neuronal death, but also these compounds that are normally produced by this pathway are not, and so this has major consequences of all neuronal functions. So we hypothesize that by bringing back the enzyme using an AAV vector delivery into the striatum, we could have therapeutic benefit for Huntington disease, and we tested that in 2 mouse models, the R62, the ZQ175. This work was mostly done by the team of Sandrine Bétuin and our lab, and in strong collaboration with Frédéric Sodou and Christian Nery that are here also. And this has allowed us to show not only correction of this mice, motor function, medium spiny neurons, and also decreased aggregates of mutant Huntingtin, but also this has allowed us to really go in depth into the mechanism of action of how restoring the brain cholesterol metabolism is indeed therapeutic for Huntington disease. So I've no time, of course, to go into details, but this slide will summarize to you what we have been able to demonstrate in vivo in mouse models. So this is what is impaired, as you know, in Huntington disease, all these numerous neuronal functions. And what we have shown is that all are restored by bringing back CYP46 and restoring cholesterol metabolism. The transcriptomic defect is corrected. The synaptic transmission is restored. The BDNF signaling is restored, and you've seen from the presentation by Fred Sodou on Thursday that the vesicular transport of BDNF can be restored, and CYP46 is doing that, and Fred has shown that. Energy metabolism is restored, and autophagy is improved. And this is the mechanism that we have demonstrated is responsible for the decrease of the mutant Huntingtin by restoring the maturation of autophagosomes. So we have decided to develop this product with AskBio. It's called BV101. It's an AV vector. It's a RH10 capsid carrying the CYP46 cDNA. It's a one-time procedure. You know that gene therapy is a one-time procedure. The vector, the DNA is not integrated but persists into cell. And it's a delivery into the striatum, caudate, and putamen using neuronavigation system. We have done dose-finding studies to demonstrate that we do have dose-dependent correction into motor behavior, in the clearance of the aggregates, and a very important marker, which is a neurofilament light chain that is restored with a dose-dependent manner. We have done translational steps in primates, starting that in collaboration with Romina Rombadin at Mersenne, and working then in a clinical setting, like you see here, with neuronavigation system that will be used in the clinics. We have demonstrated that using a vector carrying a tag in the first step, that we do have strong targeting of the striatum with also expression in the projection area, which is very important for Huntington, which is a more global disease, in the cerebral cortex and the substance nigra. So based on that, we have done tolerance studies, of course, in primates, demonstrating that not only we do have preservation of neurons, as you see here in histology, but also tolerance on the imaging follow-up of these animals. So based on that, we have decided to propose clinical application to the French regulatory agency, the ANSM. we have obtained orphan drug designation for the BV101 product. And this is an open label phase 1/2 dose finding study evaluating safety, tolerability, and efficacy of BV101 with 2 different doses. And 1 single injection into both codates and both putamins. In terms of criteria, inclusion criteria, CHG repeat expansion above 40, and total functional score of 9 to 13, and an MRI volume. As you've heard before, we decided to use volume over 2.3 centimeter cubic for putamen and 1.7 for putamen. And as exclusion criteria, I will just mention no history, of course, of gene therapy before, and no ASO delivery within the 6 months before this gene therapy trial. So it's a 3 steps: screening 8 weeks, first follow-up 52 weeks, and long-term follow-up 4 years. We will use 2 different doses and 3 patients per cohort, with 6 weeks between each patient and a very strong safety follow-up. DSMB meeting after the first and the second cohort. We will have a selection after the end of the second cohort to define the expansion phase for 6 more patients. In terms of endpoint, of course, primary endpoint is safety, and we have a lot of follow-up in terms of clinics, imaging, and biological follow-up, particularly imaging, a lot of different sequences to really document the images that, as you know, have been sometimes found in this kind of trials with intracerebral delivery. In terms of secondary endpoints, of course, the volumes of the different structures of the brain, the different clinical parameters, the NFL, the mutant Huntington, and the 24-hydroxycholesterol in blood and CSF. And importantly, because we have this metabolic approach, we think that we should be able to see some modification between before treatment and after treatment in MR spectroscopy profile, measuring different compounds, and glucose metabolism using the PET glucose before and after treatment. And of course, also we have a number of other exploratory endpoints to really go in more depth into this mechanism of action. So I will stop there. By warmly thanking first all the people that have participated to the preclinical work. It has took us more than 10 years to do all that, particularly the lab of Sandrine Bétuan, Jocelyne Caboche, and my lab at INSERM, but also, of course, Fred Sodou, as I mentioned, and Romina, who helped us for the first steps, lipid metabolism also by Antonin Lamazière. And warm thanks to my AskBio team and previous Brain Vectis team, in particularly Sandro Alves, who directed the preclinical research, and the clinical team with Alexandra Dur, that has helped us so much to set up this protocol and will be the PI for the trial. You have the contacts here. And I wish to warmly thank also CHDI and EHDN, with whom we had long-term interactions and who helped me from the very beginning to think of this cholesterol strategy and to improve this clinical protocol. So many thanks to all of you, and I hope to be back to give you more information when we start. Thanks.
Thanks a lot, Nathalie. We have time for a couple of questions. There is one over there. Please. Microphone is coming, I hope. Yeah. Good.
Thank you. That's a very interesting talk. Ruth Carter from Leicester School of Pharmacy. I was wondering how much you might have considered the heterogeneity in the data regarding cholesterol homeostasis in Huntington's disease, and the fact that this may be explained by an early accumulation of cholesterol and a subsequent downregulation of cholesterol biosynthesis. First of all, what do you think the mechanism of the CYP46A1 expression is at the cell survival level? And two, surely you're aware that 24S-hydroxycholesterol is a positive allosteric modulator of the NMDA receptor, and that there is a potential for that to exacerbate excitotoxicity locally in the highly expressing areas.
Thank you for the question. And there's a lot of things in what you asked. The first thing is what we show is CYP46 is decreased in the brain of patients and the mouse model. This is really something that we have confirmed in all models, together with this global decrease of the pathway. And what we have shown in all models is that when you restore this, you restore not only the concentration of cholesterol in the membrane, which is too elevated in untreated animals, and we restore the production of the important compounds. So our strategy is really based on the fact that CYP46 is indeed decreased. The second thing is regarding the 24-hydroxycholesterol. I think it's really a matter of concentrations, and what we have observed with all the work done in vivo and in vitro is that we decrease excitotoxicity by bringing back CYP46A1. So, and the other thing is we also did experiments in the very beginning showing that inhibiting CYP46A1 is extremely deleterious to both cells in vitro and in vivo to animal in different type of brain regions. So
I just have one maybe little follow-up point. Have you considered the possibility that the downregulation of CYP46A1 is a late compensatory mechanism?
I have more the impression that increased expression of CYP46A1 at the beginning of the disease that we have shown in some other models is a compensatory mechanism, and when it goes under a certain threshold, and because there is such a major transcriptomic defect in Huntington disease, as in other diseases with aggregated protein accumulation, there's a time where it cannot be compensated anymore. And when it goes under a threshold, then you see the toxicity, because CYP is a stress response factor in neurons, very important. This has been well characterized by other teams. And so when you cannot bring CYP to respond to stress, then you have this cascade of toxic events and that lead to neuronal death. This is our hypothesis that we have confirmed in different models and in different neurodegenerative disease, like Alzheimer's, for example. Sorry for the long answer.
This was an important discussion to have. And I don't see more questions. Maybe, Nathalie, there's one more?
Yeah.
Oh, yeah, over there. Sure, please. Quick question, one more.
Quick answer.
Quick answer.
Yeah, quick answer.
No, you did fine.
Not metabolic.
All fine.
Yeah, here. Thanks a lot for
Sure.
the nice talk. I have a question related to your autophagy mechanism. It's very interesting that you stimulate it so you will degrade the large aggregates.
Yeah.
Have you looked if soluble Huntington or small aggregates are also degraded with the gene therapy that you're proposing?
I don't think we have looked in detail in the size of the aggregates. What I can tell you is this is really a common mechanism that we have characterized on the Huntington protein, but also on ataxin-3, for example, in the model of spinocerebellar ataxia, or for amyloid. So it's really a common mechanism that we do see. But this is a good question. I think something that we could look in more detail. Thank you.
Terrific.